Hello, everyone, and welcome to Chapter 8, where we'll be talking about mitosis and meiosis. So let's jump right in. So the book likes to relate biology and society. A good example of this is recently, so in 2002, zookeepers noticed a single female Komodo dragon laid eggs. And the eggs developed with no male present. So how did this happen? How did we have viable offspring without a male? Well, the offspring only had the female's DNA. So this is called parthenogenesis. And this Komodo dragon basically cloned herself. And we're going to learn a little bit about that. So let's look at what cellular reproduction accomplishes. It can be the birth of new organisms, but generally it just means the making of new cells, especially in us. So what happens when a cell undergoes reproduction is that you make two identical cells called daughter cells. So daughter is not indicating female, it's just indicating offspring. So we just call them daughter cells. They're identical to each other and to the parent cell. So these daughter cells are little clones, if you will. And they have the same structure, organelles, DNA, etc. They're the same. So how does that happen? To get DNA to both daughter cells, we have to go through a process of multiplying DNA by 2 and giving half to each cell. So how they do that is by duplicating their chromosomes. And chromosomes are little DNA structures that are thick and condensed versions of genes, and it makes it easier to separate them into each new cell. And these two cells down here in this example picture originated from the same parent cell. They took their chromosome, they made copies of each, and sent one copy to each chromosome to the two new cells, so that once done dividing, they look like the original parent cell. So different reasons why we'd want to do cell division, for example, are replacing damaged cells. So if you cut your skin, you want to replace those cells, right? You want it to heal up and you want it to look like it did before the cut happened. And of course, we need this for growing and reproduction. Generally, cell replacement in adults would occur after getting, something like a cut, like I just said, and needing to replace cells in your organs, things like that. And then growth would occur during our developmental life stages, so before adulthood. So let's move on. There are two types of ways to reproduce. There's sexual and asexual reproduction, and both have their pros and cons. In asexual reproduction, you only need one individual. It's making exact copies of itself, so like the cloning I mentioned earlier. It's making exact copies of itself and growing these new individuals. For example, this down here is a hydra. And hydras have buds that are released to become another hydra that's exactly the same as the original hydra it came off of. And then another example is apples. The apples all came from the same original parent plant. Some more examples. So when we have individual single-celled organisms, such as this amoeba, when it divides, that's cellular reproduction, because if it divides, it becomes two genetically identical amoeba, but it's two different amoeba. Then we have regeneration of a sea star. So here, right here, this is a leg that broke off of the parent and is turning into its own sea star. So that's pretty cool. And then here we have a clipping. So putting down a leaf and growing a new plant out of it. You can do this with the tops of pineapples. So you cut the top off a pineapple planted, you should have another pineapple grow. In asexual reproduction, the parent and offspring have identical genes. So I know I've been saying clones for a while, but that's exactly what's happening. So the parent and the offspring are the same. And this is really important for asexual reproduction because if the environment is suitable and the parent's doing well in that environment, they're better off reproducing asexually, so on their own without needing somebody else to help them. That way, given that they're doing well in this unchanging environment, their offspring should do well as well. So what they would do, asexual reproducing organisms, they would go through mitosis. So mitosis is normal cell division. It'll be asexual reproduction where genes aren't mixing, and then just the growth and cell division within organisms. Sexual reproduction, however, is more complicated. So we have fertilization occurring here where we take genes from one parent, so let's say the egg, and then we take genes from the other parent, the sperm, and we go through a specialized process called meiosis, where basically egg and sperm each have half of the genes. Then when combined, you get a whole organism's worth of traits. So if you're sexually reproducing, you use meiosis for reproduction. You'll also hear it called meiosis, but I like to say meiosis, mitosis. That's how I learned it. But anyways, if you're sexually reproducing, you're using meiosis for reproduction, for making the sex cells, so the sperm or the egg. The sperm and egg are also called gametes. So gametes are sex cells. And then mitosis, sexually reproducing organisms also use mitosis, but that's what they're using for normal cell growth and maintenance. So mitosis would be for body cells, and we call these somatic cells, whereas meiosis is only for sex cells, for gametes. And examples here, we have this large structure right here is an egg, and all these little ones are sperms. In the second picture, we have a sponge that's sexually reproducing, and then this third picture here, we have a frog that's fertilizing these eggs. Now let's look at mitosis first. So this is the asexual reproduction and normal cell division within our body. So in us, in humans, mitosis happens in body cells, like I just mentioned. So different examples of body cells or somatic cells are skin, liver, basically everything except for the sex cells. So everything except for eggs and sperm. And again, soma means body, and these are our soma, our somatic cells, our body cells. Your cell goes through a cycle, or your cells go through cycles where they go through mitosis. In a human eukaryote, in a human eukaryotic cell, the genes will be in chromosomes in the cell nucleus, and that is what we'll focus on. So each chromosome is made up of DNA, and when DNA is in the form of a chromosome, it's really compact. So right here we have an example of the human genome and our chromosomes. We label them by size and number, so the longest first, so 1 is grouped together, it's a pair of chromosomes, 2, 3, and you can see how they're getting grouped together by size from largest to smallest. So 1 through 22 are our body, and then we have our gametes with the 6 chromosomes, X and Y, X female, Y male. So the number of chromosomes depends on what species it is. For each chromosome set, you see 2, a pair. This just means you get 1 from your mom and 1 from your dad. So for example, with chromosome 1, we got 1 from our mom, 1 from our dad. This is a pair, and it's together. Each of our normal body cells has 2 copies of each chromosome, so 1 through 22. So like I just said, the number of chromosomes you have depends on what species you are. Here's a good chart with examples of different species and the number of chromosomes they have. Ours says 46. That's because our 23 pairs here, so we have 23 pairs, right? With 2 each, so 23 times 2 is 46. And notice that the number of chromosomes an organism has doesn't necessarily indicate complexity of that organism. So like this little red rat down here has 102, but that doesn't necessarily mean it's more complex than a giraffe, for example, that has 30. Normally, DNA isn't visible when cells aren't dividing. So the cells on the left and right here, so left, right, those are not dividing, and we can't really see the DNA. Notice how different they look from this middle cell. This middle cell is dividing. And notice this strong outline out here. So remember, our DNA is in the form of chromosomes. You can clearly see it here and not in these two. But anyways, so the strong outline between the cells out here is the cell wall. So that's a good indication that we're looking at a plant, because animals don't have cell walls, right? So this is a plant that's undergoing cell division. So to reiterate, the DNA in a cell is packed. Let's look more at that DNA. So we have this double-stranded DNA right here, pretty thin. And what happens is we have to condense it to make it into thick chromosomes. So to condense it, we wrap it around these proteins, so these histone proteins. Then we wrap it around itself down here. Notice how it's getting tighter. And then it gets really thick. If cell is not dividing, the DNA is really thin and spread out and looks like this again. So whenever we're making a new copy of DNA, we take each chromosome and we duplicate it. And once we do this, we get these things called sister chromatids that are exactly identical. So here's chromosome 1. There's mom's copy, dad's copy. When you want to divide, you make the sister chromatid, and they're connected at the centromere. So here are sister chromatids connected at the centromere. So this is just the little middle part. And when the cell divides, one copy of the purple is going on to the first cell, and the other is going to the second. So that at the end, we'll have exactly what the original parent had. Here's a nice pic of how it works. So we have our original parent cell right here. We double it. We double the chromosome, duplicate. We get identical copies. The sister chromatids are joined at the centromere. And for each new cell, we make sure each gets one copy of the chromosome. So chromosome distribution to daughter cells. And remember, daughter does not mean female. It's just talking about the offspring cells. The cell cycle is how we make all of this work together. A cell cycle is an ordered sequence of events that goes from the cell's first formation to the cell's own division into two cells that we call daughter cells. And we have two distinct phases. We have interphase, so this is the non-dividing phase, and we have the mitotic phase, or mitosis, where the cell divides. This will also be called M phase. So we have interphase and M phase, or the mitotic phase, or mitosis. So M, mitotic, mitosis, all of that's the same. Most of the time, the cell will be in interphase, as you can see in this chart here. First, we've got a daughter cell that needs to grow. So we have G1, gap, we have an S phase, synthesis, and we have G2. During S phase, we take our original single copy of our chromosome and we double it. So an easy way to remember that is S stands for synthesis. So this is where the DNA synthesis, where the chromosome duplication happens. And synthesis means to make. So here's interphase again, and we can't see any individual chromosomes because the cell is not dividing during interphase. It's growing and it's getting ready for mitosis. So these dark spots here are nucleoli, where ribosomal RNA is made for ribosomes. So right here. Just a little dark spots you see. Alright, next. Next we are ready to go through mitosis. So we just doubled our chromosomes in interphase, and now we take all that DNA and split it in half. And then we have cytokinesis. Cyto is cell, kinesis means movement. So cytokinesis is cell movement, where the cell is actually divided into two cells. So this is where it pulls apart into two new daughter cells. During mitosis, we see a mitotic spindle form, and these fibers here is how we guide the copies. So these fibers, it's mitotic spindle fibers, is how we guide the copies of each chromosome to new cells. How we pull them apart towards different poles of the cell. So right here, one goes up, one goes down to the different poles. And eventually, the cell will pull apart and be two new cells. So now let's go through the stages of mitosis. I like to remember this as PMAT, and I'll reiterate this throughout. So PMAT, P, prophase, M, metaphase, A, anaphase, and T, telophase. So prophase is first. So pros are number one. Prophase is number one. The DNA condenses during prophase, and we start seeing it turn to dense chromosomes. And the mitotic spindle forms with centrioles going to each side, or centrosomes. Next, metaphase. So this is second. So we have prophase P, we have metaphase M. And a good way to remember metaphase is middle. So middle. This is where the order begins, where chromosomes line up towards the middle or towards the equator of the cell. So here we have that mitotic spindle, and we have these chromosomes moving towards the center, right here, towards the middle, towards the equator. Next we have anaphase, so PMAT, this is the A, anaphase, and this is where the chromosomes get pulled to each side, so to the different poles. They're getting away from the equator, they're getting pulled to the different sides of the cell. So it looks like they're getting dragged by the centromeres, and that's because they are getting dragged by the centromeres. And a good way to remember what's happening here is anaphase, A-N-A. The A and the A are separated by the N, so that's the A and the A are getting pulled apart. The chromosomes are getting pulled apart. And then lastly, we have telophase and cytokinesis, where chromosomes are separated to each new cell. The DNA goes back to the nucleus and it starts to string out again, so it becomes thin again. It's not thick and condensed anymore. And we get cytokinesis, where you can see it's called a cleavage furrow right here forming, and organelles are splitting up, cells moving apart, and we're going to get two new cells with cytokinesis. Cytocell, kinesis moving, so cell moving apart, two new cells. So I just mentioned cleavage furrow. That's the ring here that's forming. And what happens is this ring contracts and pinches off, and that's how the two new cells are made. So Here, the cytoplasm divides, fluids divide, organelles divide, both these two new cells. And it's actually a little bit easier to look at plant cells because, again, like I mentioned earlier, they're divided by cell walls, so you can definitely see like each individual cell more easily, I think. After the cell plate is fully formed, you get new cell walls with mitosis in plants, and that's about all I'll mention there. Here we have a picture of an onion root that's dividing and rapidly growing. And an onion root is a really good example to look at, especially in labs, because you can definitely, see like these different separate cells that are very easily divided into these squares. But anyways, each square is the cell, and on your own, try to label A, B, C, D into what you think is happening. So remember PMET, prophase, metaphase, anaphase, telophase. And try to do that with the donuts too, right here. So if you want to do that, pause the video. If you don't, then just keep listening because I'm going to go through it. In A, here we have metaphase happening. You can see the chromosomes are in the middle, so metaphase middle. And B, we have anaphase. They have been pulled to the sides, to the different poles. In C, we have telophase happening. And then in D, we have prophase, so you can't really see what's going on, but you can see that the DNA looks like it's condensed into chromosomes because they look pretty wiry right here. And then with these donuts here, we have interphase. So remember, our cell stays in interphase the longest. Then we have prophase right here. We have metaphase where they line up in the middle. We have anaphase where they start getting pulled apart. We have telophase where it starts looking like it's about to split up. And then we have cytokinesis where there are two new cells. Alright, so let's talk about cancer cells a little bit. So normally our cells have a control system that tells them when to divide and when to stop. If part of the cell division system gets out of control, that's what we call cancer. Normally cells in an area, if they're close to another cell, it'll tell them not to divide. But with cancer, that stop sequence has been turned off. So now those cells divide rapidly into other areas, and those areas of growth, those masses, are called tumors. And if the tumor stays where it originated, it's called benign, so it's not spreading. And this is usually the type of tumor you would hear somebody go to get surgery to get removed. However, if it spreads to other areas, that's called metastasis. And that would be malignant. So mal means bad. This is a malignant tumor, and these spread. And how they spread is through the lymphatic system. So if someone has a malignant tumor that is spreading, that's what most people think of as cancers, and that's what we call cancer. An example here is breast cancer. It generally goes to lymph vessels, which spread to other areas, like our lymph nodes. And our lymph system is what collects fluid and proteins from our blood, and it returns them back into our blood flow. And it's part of our immune system to help make sure that nothing gets out of hand. But if the immune system is overwhelmed, these cancer cells can travel through the lymph tissue and to other areas, which is not what we want. With cancer treatments, we want to disrupt cell division, and if we can target those cells, they won't divide, and our immune system can go through and take care of those cells. Our immune system does recognize that there's something off about those cells, and it wants to fix them. Radiation therapy, how it works. So, which damages DNA and disrupts cell division. Here's an example of how radiation therapy works. So, a surgeon implants transmitter into the prostate, and then, well, okay, so this is specifically a prostate example, and then an X-ray plate is put over the body to deliver that therapy, that radiation therapy. And then a signal ensures the radiation hits its exact target. And then we have chemotherapy, which is the use of chemicals to disrupt this. So chemotherapy prevents blood vessel growth, it stops cell division, replication, it causes cell death, and then it can create a non-dividing cell by altering the cell structure. So chemotherapy is use of chemicals, chemo, and radiation therapy is the use of radiation, as it sounds. So these are just some things that you can do right now to decrease your risk of getting cancer. This is just a list of suggestions that I will let you read through on your own. So next, let's talk about meiosis, sexual reproduction. This only, again, happens in our gametes, our sex cells. And the sexual reproduction is complicated. Asexual was so simple, but here with sexual, we have a few things we need to do. We have to find a mate, a good mate with good genes, and it's pretty hard work to do that. So where lies the benefit in this? So the benefit is in such variable traits that happen during fertilization. For example, this is a fun little picture of two twins. Obviously, they're not identical, but they have the same parents. In sexual reproduction, a benefit for this variable traits is if one is living in an environment that is less stable. So remember, asexual is great in a stable, unchanging environment. But if the environment has lots of parasites or viruses, you want variation in your offspring because some might be better suited for that environment. Do we know what'll happen? No, we don't. But that's where natural selection comes in, and hopefully some of your offspring will be able to make it in that new environment. So what we have to do is cut the number of genes we're sending through in half. So we've got 46 chromosomes, 23 pairs, one set from our mom, one set from our dad. And for each chromosome, we've got a copy, again, from mom and a copy from dad. And when a cell divides, they make a second copy. Those are called the sister chromatids. And when you look at both copies of your chromosome, so again, one from mom, one from dad, these are called homologous chromosomes. So homo means same, so they're the same chromosome. All the same genes code for the same things. So with blood type, there could be different versions. Like one side would say A blood, the other side would say B blood, but it's still blood. So remember, we have 22 normal body pairs, like we mentioned a second ago. These 22 normal body pairs we call autosomes, and then the 23rd pair is our sex cells. The X and the Y are the 23rd pair. So what we do is go through meiosis. As normal body cells, we've got, again, 46 chromosomes. I know it gets a little repetitive, but through repetition, we learn things, right? So, anyways, we've got 46 chromosomes. We've got what's called 2N diploid. So, remember, 2 times 23 is 46. And it just means we've got 2 copies of each. So what happens when we go through meiosis is we only get 1 copy of each. So we'll get mom's chromosome 1, dad's chromosome 2, dad's chromosome 3, just for example, instead of getting 1 from mom, 1 from dad for each. And then those egg and sperm that each have 1 copy combine together to make this fertilized new organism that's diploid. So that's this one. It's got 2 copies of each chromosome again. And then through mitosis, we go to our regular developing into adults form. So this is all meiosis, and then mitosis for development. So we are diploid. We've got two sets of chromosomes, whereas our sex cells are haploid, and they only have one. So during meiosis, our goal is to get down to only one copy of each chromosome. Then our gametes, the sperm and egg, fuse together and form a new cell called a zygote. So the zygote is when the sperm and egg fuse together. And that starts going through divisions to become new life. So that zygote turns into a baby, turns into a toddler, turns into a teenager, then a human, and not human, then an adult. That's funny. All right, next. Here's our normal body cell. For example, chromosome 1. Mom's copy, dad's copy. Before any division, we duplicate during S phase, so we duplicate the chromosomes. During S phase and interphase to make 2 copies of each. So what happens in meiosis 1, the homologous chromosomes separate. And now we have 2 daughter cells that are not identical to the parents. They're diploid. During meiosis 2, we don't make second copy of DNA, and we separate out so that we have 4 non-identical cells with 1 copy of each chromosome. In humans, if this was sperm, each of these right here, would become a sperm cell. If they were eggs, only one of these 4 would become an egg. And what's cool in humans is that meiosis has already started on eggs before birth. They get to a point, they stop, then during puberty they go through the rest of these steps during ovulation. That's why we say eggs are expensive and sperm are cheap, because all of these will turn into sperm in a male, but only one would turn into an egg. So viable egg in a female. What happens is we end up with 4 haploid cells with only 1 copy of each chromosome. What's super cool is that crossover occurs during meiosis, and although both of these right here are yellow, they are not identical. Crossover happens, in this example right here, when homologous chromosomes are touching and a part of each chromosome gets switched. So here they're touching, get switched. The chiasma is what it's called, where that switching occurs. And you can see how each have a little bit of information from the other. So meiosis 1, we separate out homologous chromosomes and crossover occurs. And in meiosis 2, we separate sister chromatids. Same copy of chromosomes, except they have some crossover, and the result is 4 different cells. So let's review a little bit. Mitosis is for body cells and making identical copies of the parent. And then meiosis is for sex cells, for gametes. So mitosis, body cells, somatic cells; meiosis, sex cells, gametes. In both of these, the chromosome duplicates just once during S phase and interphase, but the number of cell division varies. So during mitosis, we have 1 division that produces 2 diploid cells, and in meiosis, we have 2 divisions that produce 4 haploid cells. So this is a nice picture to review with. We have, in mitosis, we have PMAT. In meiosis, we have PMAT1 and PMAT2. So meiosis 1 and meiosis 2. And they're very similar, except that meiosis, you get genetic variation and crossing over, and that crossing over is important for genetic variation. And in mitosis, we get 2 daughter cells that are diploid, and in meiosis, we get 4 daughter cells that are haploid. So next, the origins of genetic variation. Offspring of sexual reproduction are genetically different from their parents and one another. So this is where that genetic variation steps in. Because of variability, some offspring can be more successful than others. For example, the spotted pattern could make this one more easily visible to predators, whereas this dark coloration makes it less visible and more elusive. And this is where natural selection would come in. So if he can hide better, he's not going to get eaten by a predator, right? Or vice versa. Maybe the spotted pattern breaks up the deer and confuses the predator. I don't know. It's where natural selection steps in. So independent assortment of chromosomes theory states that picking which homologous chromosomes go to which new cell during meiosis is random. So there's 50-50 chance for each. So here are different possibilities, but in both there's 50-50 chance for blue or red, right? It happens randomly. This randomness or variability helps increase the amount of possible variations on offspring of sexual reproduction. And this example here is of Mendel's pea plants, which we'll look at in the next lecture, and of all the different possible variations that can happen. We just discussed crossing over, but let's touch on it again. So crossing over, variation, genetic recombination. This is just, genetic recombination is just talking about all the different gene combos you can have sent from parent to offspring. So some traits are linked. This means they won't get separated during crossing over. And different examples of different linked traits are things like light hair, light eyes, dark hair, dark eyes, things like that. This shows a really nice picture of crossover. So we've got our homologous chromosomes at the top, and we're going into meiosis to make sex cells. We've doubled our DNA, so during meiosis 1, these line up at the middle. Material exchange information. And then during anaphase, they get pulled in different directions, except they've exchanged some DNA. Then after meiosis 2, we pull apart each of these, and we've got the 4 different possible options, 4 different possible outcomes. And then these are the recombinant chromosomes, where we've got part of the DNA of one chromosome mixed with the other. There can be errors. Oftentimes, if there are errors, these can be fatal. So first, let's look at non-disjunction. This is where chromosomes aren't separating properly, so offspring have the wrong number. So they either have one too many or one too few. This is fatal almost all of the time. If you get 3 copies of a chromosome during development, the zygote will be aborted. So this can either occur at meiosis 1 or meiosis 2. If this shows an egg with an extra copy of a chromosome, and this is what would be called diploid plus 1 extra. Down syndrome is the only non-disjunction that is not lethal other than with our sex chromosomes. So here we have an extra copy of chromosome 21. So we have 3, so trisomy 3, chromosome 21. Right there, 3, 1, 2, 3, 3 copies. This is a table with sex chromosome abnormalities. So we have Klinefelter, where we have an extra X, And this is example A, Klinefelter syndrome, where we have smaller testes, more female-type pubic hair, breast development in a male. And we also have Turner syndrome right here, XO. So this is an example right here in B. And this person only has one X and is generally shorter in stature and infertile. So let's review some different advantages of sexual and of asexual reproduction. So asexual reproduction is good when it's in a stable environment, in a non-changing environment, and it also eliminates the need to spend energy finding a partner, making gametes, making sex cells, copulating with a partner. So animals are also oftentimes at their most vulnerable when they're fertilizing eggs. So most are vulnerable to predation, which would be another handy thing for asexual reproduction. Here's an example of asexual reproduction in strawberries. Strawberries send out these runners, which are asexual identical copies. They're making seeds with the strawberries, and think about how much energy is saved there. They can expand from their area without too much work. So now we have advantages of sexual reproduction. So that work that we were just talking about, asexually reproducing species saving, so all that energy they've saved, it can actually be very good. It can be very advantageous with sexually reproducing species because it gives them genetic variation, which we just talked a lot about, to help with unstable environmental change. So remember, it's not thinking. It's not going to think, oh, it's cold, let me put on a jacket. It doesn't do that. It allows a population to get rid of harmful genes. So if you're having trouble with meiosis, I suggest finding more videos online or notes in your book, flashcards in MyLabs Plus. There's some stuff in your homework that will go over it, but it never hurts to find more study material. And that is all for Chapter 8. I know it's a long one, but it has a lot of good information. All right, and that's it.